going to look green. So when we're given information about wavelength and light absorption, we can start to apply that to how that will affect the rate of the photosynthesis. Okay, so for example, up here on the top, I have what's called an absorption spectrum, and that's what we were just looking at. Okay, so that absorption spectrum is giving me information about what wavelengths of light are absorbed, right? Relative absorption for the various wavelengths of light. And then below it, I have what's called an action spectrum. Okay, and so that action spectrum is giving me information then about the effectiveness, so the rate of photosynthesis, right? Photosynthesis would be higher over here at the violet blue wavelengths of light, as well as the orange and the yellows, because that's where I'm able to absorb the most light on my absorption spectrum. So the more light I can absorb, okay, the faster photosynthesis will be happening. So if you had different color light bulbs, different color lights available to you, like you were only, like for instance, if the plant was only exposed to these green wavelengths of light, it may not survive. It may not be able to do enough photosynthesis to be able to survive. So you can kind of put those two and two together and apply your absorption spectrum to form an action spectrum. Let's do a little bit of photosynthesis review here. Okay, so if you all remember hopefully what comes into photosynthesis, right? I've got water, I've got carbon dioxide, I need light energy, and that's going to give me oxygen and my organic compound, which usually I'm going to talk about with glucose. Okay, your freshman year, we didn't do too much with trying to balance this equation or anything. This year, you'll probably see a lot more of the balancing of the equation. Okay, so remember to balance it, I'm just putting my 6's in front here, and now I've got my balanced equation. Okay, you'll never be asked to balance an equation in this class, so I'm not even going to worry about whether you can do that. We're going to let Ms. Wallace worry about whether you can do that or not. Okay, so this reaction is an endergonic reaction. Okay, so remember endergonic reactions, this requires energy input. Okay, so this is going to require an energy input here, and that input of energy is going to come in the form of light. Okay, so here's my energy input, so that's what's going to make this an endergonic reaction. And again, it's got two stages. Okay, it's got the light reaction. Okay, so it's got the light reaction as well as what's called the Calvin cycle. You may occasionally hear them call the dark reaction, although I seriously doubt you would see it on your test, or anybody that's really knowledgeable anymore would really call it that. But you may see light independent reaction. And I can't spell today. Let's try to spell again. Okay. So if you see light independent reaction, that is the same as the Calvin cycle. Okay. One of our main focuses as we go through this is going to be the energy. Where is the energy going? Okay, where is it? Because remember, we're not creating it, we're not destroying it. So what is the energy flow that's happening here? Okay, and so let's just kind of get our big idea under our belts here. So the energy is going to start with the light energy coming into the reaction, right? That light energy is going to be converted into ATP and NADPH. Okay, that's going to happen during the light reactions. Okay, so that's part of what's happening there during the light reactions. And then that energy is going to be converted into the organic compounds. And that's what's happening during the Calvin cycle. Okay, so remember, we're not creating, we're not destroying any energy here. We're just converting it. And eventually it will end up in that chemical or stored. So let's focus on the light reactions now. This is where your stuff that's really going to show up that's brand new. That shouldn't really be reviewed from freshman year. We're going to get into some stuff that is going to be brand new here. Okay, so the light reactions are going to make use of what are called photosystems. Okay, so your photosystem is going to be basically a cluster of pigment molecules. So you've got all these little green circles here. These are your pigment molecules. So you've got a cluster of pigment molecules bound to proteins, and there's a primary electron acceptor. Okay, so we've got basically what you need to realize here is I've got some pigments, some proteins, and an electron acceptor. Okay, so all those things combined here are going to make up my photosystem. And I've got two photosystems here. Okay, so I've got what's called photosystem 2. And yes, I know I'm out of order here. Photosystem 2, which is sometimes called P680. We won't really refer to it as that. You may see me abbreviate, though, as photosystem 2. Okay, and then we also have what's called photosystem 1. Okay, which is P700, and those are the wavelengths of light that they absorb the best. Okay, so photosystem 2 works best at 680 nanometers, photosystem 1 works best at 700 nanometers, and you may see this one abbreviated as PS1. Okay, so they are named not for the order that you're going to see them in these reactions. So you're actually going to see photosystem 2 first, and then photosystem 1. They're named based on the order they were discovered. So photosystem 1 was actually discovered first, and photosystem 2 was discovered second. Don't ask me why we didn't change it or give them a better name or anything like that. Just like all biology things, I guess we just wanted to keep it confusing. So we're going to start off with photosystem 2. Okay, so let's take a look at what's called non-cyclic electron flow. Okay, so what I have happening here is non-cyclic electron flow. Okay, cyclic would be a cycle, right? So this is not cycling. We're going to have non-cyclic electrical flow, and then you'll see cyclic electrical flow. Okay, so this one, the electrons are not going in a continuous cycle or circle. Okay, and so don't let the picture freak you out. Okay, don't let any of this, stress you out. You're not going to have to memorize every single little detail. Okay, what you need to get is we need to get a handle of the energy flow and what molecules we're helping, that energy flow is helping form and create. Okay, so what we're going to do is we're going to start off here, okay, with photosystem 2. Okay, so I've got photosystem 2 coming into play with this one. And so photosystem 2 is going to absorb the light energy, because remember the photosystem is made up of all of these pigments. So it's absorbing the light energy from the photon. Remember the photon is the light. Okay, so photosystem 2 is absorbing the light energy from the photon. And when it absorbs that light energy from the photon, it excites one of its electrons. So the electron was really down here at this energy state. And the photosystem, so this electron was already in the photosystem. Okay, the photosystem absorbed the light energy, and that made the electron get really excited. Okay, so the energy of that electron went up. Okay, well now the photosystem has a hole in it. Okay, it's missing an electron. So the photosystem needs to fill that electron from somewhere, and that's where water is going to come into play. Okay, so the electron got excited and left the photosystem. Now the plant is going to use water to replace the electron that left. So the water will be split. Okay, so the water is going to be split using what's called photolysis. Okay, so we're using that light to help split the water. Okay, so the water is going to be split, and it's going to release oxygen. Okay, and it's going to release these hydrogen ions. Okay, because remember, hydrogen normally, right, if I have hydrogen, okay, hydrogen has one proton, right, and one electron. Okay, so when I split the water, okay, I'm going to take the electrons off of the hydrogen and use them to replace the ones that went missing from photosystem 2. And so now I have left over, I've got H+, so I have hydrogen ions, okay, and I've got oxygen. The oxygen's going to be released. The oxygen's going away. It's going to be released from the plant. Okay, the hydrogen ions, they're going to be used. They're going to be extremely important. They're going to be used to build a concentration gradient. Remember we talked in the beginning about how important that membrane was so that we can build the concentration gradient? And if you think about where we are, we're on the thylakoid membrane right now. These photosystems are embedded in the thylakoid membrane. So these hydrogen ions are going to be shuttled, okay, to one side of the membrane to help create a concentration gradient. And we'll talk more about the point of that and what that's going to do in a little bit. Okay, so So now I've filled the hole in the photosystem 2. So photosystem 2 is happy, but I got this electron hanging out, this really excited electron up here. And so that really excited electron is going to start traveling down the electron transport chain. Okay, so I've got this electron transport chain here. And so this very excited electron is headed down this electron transport chain. Okay, and it's going to be able to head down the electron transport chain because each one of these proteins here, each one of these complexes in the electron transport chain is more electronegative than the previous one. Okay, it doesn't matter if you know their names. You do not need to know PQ and cytochrome complexes. Okay, you just need to know that this one down here is more electronegative than this one up here. Okay, and by electronegative, we mean it has a stronger ability to attract electrons to it. So the electron is going to move down here because each one of these molecules has a stronger affinity for electrons than the one before. So they're going to be pulling it through the electron transport chain. And as it gets pulled through the electron transport chain, it's going to quote-unquote lose energy. Remember, it's not going to totally lose it because we can't create or destroy energy here. But it's going to release energy that is going to be then harnessed for that proton, for that concentration gradient. Okay, that energy is going to be used to help form this concentration gradient that we were talking about over here with the hydrogen ions. So we're not truly losing the energy. It's all part of a coupled reaction. Okay, so when reactions are coupled, that means I've got one reaction that's releasing energy and one that needs an input of energy, one exergonic and one endergonic reaction. And so I'm releasing the energy here, and then I'm harnessing it for that concentration gradient. Okay. All right, let's get a clean picture, and we're going to keep going with this. Okay, so I have a clean picture, okay, and so let's keep going with what's happening here. Okay, so I've got my electron has made it, so my excited electron from photosystem 2, okay, has gone here. It's made it down the electron transport chain. Okay, so the hole in photosystem 2, remember, it got filled by the water. Okay, the oxygen that came from the water splitting leaves the plant. The hydrogen helps make the concentration gradient. And you can see here, remember we were talking about that coupled reaction, the energy flow that is the energy that's being lost from the electron as it's traveling through these more electronegative molecules is harnessed to create ATP to help with the concentration gradient. We'll talk about that next. So we also have, though, photosystem 1. Photosystem 1 hasn't really done anything yet. Okay, so photosystem 1, okay, the light energy is also going to impact photosystem 1. Okay, it's going to have that same instance where the electron is going to get excited. Okay, here's my energy of my electrons, so the energy of the electrons gets raised to a higher state. Well, just like photosystem 2, photosystem 1 now has a hole. It's missing an electron here. Well, the electron from photosystem 2 is what fills that hole. You'll notice there's no water over here with photosystem 1 because photosystem 1 fills its missing electron hole with the electron that got excited and left photosystem 2. So this is what we mean by non-cyclic electron flow. It's not, the electron isn't going from photosystem 2, making a cycle and coming right back to photosystem 2. It's going from photosystem 2 to somewhere completely different. Okay, it's headed over there to photosystem 1. And so the electron that got excited at photosystem 1 got excited. Okay, it went to its electron acceptor, and it's going to travel down its own electron transport chain. And as it travels down its own electron transport chain, okay, it's going to travel down there, quote-unquote, losing energy, and is eventually going to reduce NADP plus gets reduced to NADPH. And NADPH serves as an electron carrier, and that's going to be important for the Calvin cycle. So that NADPH is required to go over to make the reactions of the Calvin cycle work. Okay, so now let's take a minute there. So we've talked about all these electrons. We've talked about where the electrons have gone, how they've left their photosystems, and they've traveled down the electron transport chains. Okay, but we also were mentioning that this hydrogen was creating a concentration gradient. And as the electron went through this first electron transport chain here, it was providing energy to make ATP. So let's back up for a second and take a look at how that is going to work. Okay, so just a quick reminder here of what we're trying to make. Okay, so here's our ATP. Remember, ATP is made of an adenine molecule, adenine nitrogen base. okay, a ribose sugar, and 3 phosphate groups. Okay, and so ATP is able to do cellular work. It is considered an unstable molecule because of